We've defined tissues as being made of cells working together to perform a specific function. But there's more to a tissue than just the cells. A tissue involves the cells and the extracellular matrix that's around the cells. The extracellular matrix is what fills the space between the cells. The extracellular matrix is made of two things. We have protein fibers. These protein fibers are secreted by the cell and released into the spaces in between them. And the ground substance. The ground substance is the fluid that fills the spaces between the cells and is all around these fibers. This is also secreted by the cells. The proteins of the extracellular matrix form a network that fills the spaces between the cells. The main fibers that are involved in the extracellular matrix are collagen fibers, which are long, thick fibers. These fibers are flexible but not very stretchy. And elastic fibers. The elastic fibers are thinner and not as strong as the collagen fibers, but they are stretchy, elastic, so they allow tissues to have a little bit of stretch or give to them. The ground substance that we find around the cells and around these fibers is important because it is the medium through which a lot of important things travel from cell to cell. This ground substance contains the nutrients, wastes, and signaling molecules that are going from one cell to another. The consistency of the ground substance is important to the function of the tissue. In some tissues, the ground substance is very watery. In others, it's a little stiffer, sort of gelatinous. In tissues like cartilage, the ground substance is actually fairly rubbery and somewhat stiff. And in bone, the ground substance is actually hard. Especially in tissues like bone and cartilage, the extracellular matrix is as important to the function of the tissue as the actual cells. Other structures that contain more extracellular matrix than cells would be tendons and ligaments, and even the cornea of the eye. Before we get too far into looking at different types of tissues, I want to talk a little bit about tissues in general and how we're able to view them. We see tissues using a microscope. But we can't see an entire big chunk of tissue at one time using a microscope. In order to see tissues, we have to take a chunk of tissue and then we have to fix it, which is what keeps the whole thing from falling apart while we're working with it. And it's often embedded in wax and then sliced very, very thinly. This gives us the thin little slices of tissue that can go on a microscope slide that we can actually look at under the microscope. One of the things to consider about a tissue is the direction that it's going to be sliced. Depending on the direction that a tissue is sliced, it will end up looking quite different. Imagine something we can see easily, like a tree trunk. If you were to take a big log, a big tree trunk, there are a couple of different ways that you could slice it. You could slice it acrossways, that's called a cross section, and then you would see round circles with rings. But if you took that same section of a tree trunk and you sliced it longitudinally, that's the long direction, then you have something that looks like the wood grain in a board. It's the exact same structure that you start with, but depending on how you slice it, it can look different. This is true of tissues as well, and it's important to know which direction a tissue is being sliced in order to understand what you're looking at. In addition to the direction that the tissue is cut, we also need to be concerned with how the tissue was stained. Most body tissues are transparent in thin sections. That makes it really hard to see the cells and the features of the cells. So most tissue sections are stained before they're viewed. Using different combinations of dyes, we can color the cells so that we can see them. So when we look at tissue slides, we can see various colors of purples, blues, reds, blacks, and pinks. Natural tissue does not have those colors. Your cells don't have bright red or blue nuclei, but we add the stain so that we can see the features more clearly. It's important to know that because the color of the tissue itself doesn't really tell you what the tissue is. It just tells you what was used to stain the tissue. We're going to be investigating the 4 basic tissue types. The 4 basic tissue types are epithelial tissue, connective tissue, muscular tissue, and nervous tissue.